For this chapter, we will be talking about synaptic transmission. A synapse is a special junction where a part of the neuron contacts and communicates with another neuron or cell type. The neuron passing the information across its axon is the presynaptic neuron, and the target cell which receives the information is the postsynaptic cell. The information transfer at the synapse is defined as synaptic transmission, and this mostly occurs in one direction, from the neuron to the target cell. Synapses can be differentiated by whether it is an electrical synapse or a chemical synapse. Electrical synapses are generally fast transmissions that can be bidirectional. It is common in several parts of the mammalian central nervous system, such as the retina and hippocampus. Because of the fast transmission that electrical synapses entails, they are usually involved in rapid responses, such as defensive reflexes. For example, in the crayfish, electrical synapses are found between sensory and motor neuronal pathways, which regulates its escape from danger, facilitating its retreat attempts from any threats it faces. Electrical synapses occur at specialized sites called gap junctions. Every gap junction consists of numerous gap junction channels that allow ions to pass from one cell to another, transferring information in the process. The following diagram shows a close-up view of the gap junction between two cells. The gap between the two cells are vastly populated by small proteins known as connexins. 6 of these connexin subunits can combine to form a channel known as a connexon. When two connexons meet and combine, a gap junction channel is formed. Cells connected by gap junctions are known to be electrically coupled. These gap junction channels have large pores in them of about 1 to 2 nanometers in size, which is of sufficient size to allow ions to pass through them. These channels also allow ions to pass through them in both directions, giving rise to the bidirectional characteristic distinctive of electrical synapses, unlike chemical synapses which are monodirectional. When two neurons are electrically coupled, an action potential that occurs on a presynaptic neuron can result in a small amount of ionic current to flow into the postsynaptic neuron. This results in the emergence of a postsynaptic potential, PSP. The reverse can also occur for the presynaptic neuron, as the transmission can also be bidirectional. So when the postsynaptic neuron generates an action potential, a PSP is also generated on the presynaptic neuron. As a neuron usually synapses onto several other neurons, several PSPs occurring simultaneously can result in a very strong excitation for the neuron. This phenomenon is known as synaptic integration, and it will be touched upon later on in the video. The following diagram illustrates the components of a chemical synapse. Both the presynaptic and postsynaptic ends of the synapse is separated by the synaptic cleft. This synaptic cleft is a lot larger than the size of gap junctions, and it averages between 20 to 50 nanometers. This cleft is filled with extracellular fibrous protein that acts as a glue to hold both the post and presynaptic ends together. The presynaptic site is called the presynaptic element. Inside it are numerous spheres known as synaptic vesicles. These vesicles contain neurotransmitters that are used to communicate with the postsynaptic neuron. Bigger vesicles are called secretory granules and they contain the larger peptide neurotransmitters, which will be mentioned later. The accretion of proteins within membranes of both sides of the synaptic cleft are the membrane differentiations, which encompasses the active zones and the postsynaptic density. The active zones are the sites in which neurotransmitters are being released on the presynaptic end, while the postsynaptic density houses the neurotransmitter receptors, which convert the extracellular signal, which are the neurotransmitters, into an intracellular signal, such as the inducing of membrane potential changes on the postsynaptic cell. The chemical synapses can be differentiated based on which part of the neuron is postsynaptic to the presynaptic axon. In the figure here, for A, it is an exodendritic synapse, which is where the postsynaptic membrane is on a dendrite. In B, it is an exosomatic synapse, where the postsynaptic membrane is on a cell body. For C, it is an exoaxonic synapse, where the postsynaptic membrane is on another axon. On top of that, there's also the exospinous synapse, which is a presynaptic axon contacting a postsynaptic dendritic spine, and dendrotritic synapse, where dendrites form synapses with one another. They can be further classified to gray type 1 or type 2 synapses based on their membrane differentiations. Those with thicker postsynaptic membrane differentiations are gray type 1, which are asymmetrical synapses. These synapses are usually excitatory by nature. Those with similar membrane differentiation thickness are gray type 2, which are symmetrical synapses and are usually inhibitory by nature. A particular chemical synapse, where the axons or motor neurons of the spinal cord synapses onto the skeletal muscle, is known as the neuromuscular junction. This synapse is very fast and reliable due to a few reasons. Firstly, there is a large number of active zones present at the presynaptic end. This means that there is abundance of sites available for neurotransmitter release. The motor end plate at the postsynaptic region has many shallow folds that comprises of many neurotransmitter receptors. This ultimately results in a large amount of neurotransmitters being released onto a membrane surface that is highly sensitive towards these neurotransmitters. An action potential in the motor axon will almost always generate another on the muscle cell that it synapses on. Neurotransmitters are broadly categorized into three main categories: amino acids, amines, and peptides. This table below highlights some of the members in each of the three categories. Amino acids and amines are generally smaller neurotransmitters that are stored in the synaptic vesicles, while peptide neurotransmitters are larger and are stored in the secretory granules. These peptide neurotransmitters tend to regulate slow and ongoing brain functions. Most central nervous system synapses are mediated by amino acids, glutamate, GABA, which is gamma-aminobutyric acid, or glycine. On the other hand, ACH, which is acetylcholine, mediates the synaptic transmission at neuromuscular junctions. Neurotransmitters are synthesized in many different ways. For example, glutamate and glycine are members of the 20 amino acids that are the basic foundation to build proteins. As a result, they are abundant across the cells in our body, including neurons as well. On the other hand, GABA and amines are made by the neurons that secrete them. These neurons contain specific enzymes that are able to synthesize them from several metabolic precursors. The peptide neurotransmitters are different in the way they are synthesized too, as the synthesis occurs in the rough endoplasmic reticulum. The following diagram illustrates the two pathways in which the neurotransmitters are synthesized. For the smaller amino acids, the enzymes convert the precursor molecules into neurotransmitter molecules in the cytosol. This is followed by the assistance of transporter proteins that aid in allowing the synaptic vesicles to uptake these neurotransmitters and store them. For peptide neurotransmitters, the synthesis begins in a rough ER where the synthesized long precursor peptide is being split in the Golgi apparatus to give the active neurotransmitter. The secretory granules then split off from the Golgi apparatus and are carried to the presynaptic axon terminal by exoplasmic transport. The diagram here illustrates the process of exocytosis, which is the release of neurotransmitters. The process kickstarts itself when there is an action potential at the axon terminal. The depolarization causes the voltage-gated calcium channels at the active zones to open. Similar to sodium channel explained in the action potential video, this results in an influx of calcium ions that will flood the axon terminal. The signal for neurotransmitter release is achieved as calcium ions flood the cytoplasm of the axon terminal. This leads to the synaptic vesicles releasing their stored neurotransmitters into the synaptic cleft. This process occurs very rapidly, within 0.2 milliseconds of the entry of the calcium ions. Peptide neurotransmitter release that isn't depicted in this diagram is also calcium dependent. However, this release is slower as the secretory granules are not located at the active zones where the voltage-gated calcium channels open. This release requires high-frequency action potentials, so the calcium levels at the axon terminal can be elevated to a high enough level to trigger its release. This process takes a much longer time, averaging 50 milliseconds. This video here will illustrate the process of chemical synapses.
A chemical synapse is a special junction between two neurons that allows them to communicate without being physically connected. A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft. An action potential causes voltage-gated calcium ion channels to open in the presynaptic terminal. The influx of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis. This neuron is releasing acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
Release of neurotransmitters into the synaptic cleft will affect the postsynaptic neuron by binding to the receptors at the postsynaptic density. The two types of receptors that can be classified to transmitter-gated ion channels and G-protein coupled receptors. Transmitter-gated ion channels consist of 4 to 5 protein subunits that combine to form a pore. This pore is usually closed when no neurotransmitters are present. However, when one binds to a specific site on the channel, a conformational change occurs and the pores open. This results in the channel allowing ions to pass through. For example, sodium ions can pass through and depolarization of the postsynaptic cell occurs. This could lead to bringing the membrane potential to threshold levels to generate an action potential. As such, the effect is described to be excitatory. The reverse can occur if the channel is permeable to chloride ions, which the net resulting effect will be to hyperpolarize the membrane potential, and thus this effect can be described as inhibitory. The other form of receptor is the G-protein receptor that is activated through 3 steps. Firstly, neurotransmitters bind to the receptor proteins that are at the postsynaptic membrane. These receptor proteins proceed to activate small proteins known as the G-proteins that have the freedom to move along the intracellular phase of the postsynaptic membrane. These G-proteins then activate effector proteins. These effector proteins can be G-protein-gated ion channels that can open when G-proteins activate them, or they can be enzymes that synthesize second messengers. Second messengers act to activate other enzymes that play a role in cellular metabolism and regulation of ion channel function. The figure here depicts the generation of an excitatory postsynaptic potential, EPSP. This was described briefly 2 slides earlier on transmitter-gated ion channels. Essentially, it begins with the release of neurotransmitters at the presynaptic end, and the neurotransmitters induces a conformational change at the ion channels, causing them to open. In this case, the channel is permeable to sodium ions, and the sodium ions are able to enter the postsynaptic cell, causing a depolarization of the membrane potential, giving rise to an EPSP. This is notably caused by ACH-gated or glutamate-gated ion channels. For the other figure, the reverse occurs and chloride ions enter instead, leading to hyperpolarization of the membrane potential, giving rise to an inhibitory postsynaptic potential, IPSP. IPSPs are caused by the activation of glycine-gated or GABA-gated ion channels. After interaction with the postsynaptic receptors, neurotransmitters are cleared from the synaptic cleft to be made ready for another round of synaptic transmission through a few different ways. One of the ways this occurs is through diffusion of the neurotransmitters through the extracellular fluid away from the synapse itself. Another way this occurs is through the reuptake by the presynaptic terminal through the action of neurotransmitter transporter proteins. Following that, they are either loaded back into the synaptic vesicles or degraded by enzymes and the waste products recycled. The third way is through terminating the neurotransmitter action by enzymatic destruction at the synaptic cleft. This is particularly important for acetylcholine at the neuromuscular junction. The enzyme acetylcholinesterase cleaves acetylcholine, inactivating it at the acetylcholine receptors. This is important as if the removal mechanism is not present, the constant exposure to high amounts of acetylcholine can lead to desensitization of the transmitter-gated channels. This means that even in the process of acetylcholine, the channels will not open, rendering the failure of neuromuscular transmission. Neuropharmacology is the study of drugs' effects on our nervous system tissue. This study is broken down into two ways in which the drugs can act: receptor antagonists and agonists. Receptor antagonists are inhibitors of neurotransmitter receptors, while receptor agonists are drugs that imitate the effects of the natural neurotransmitter. An example of a receptor antagonist is curare. Curare is a poison applied at the tip of arrows and darts by South American natives to paralyze prey. Curare acts by binding to acetylcholine receptors on skeletal muscle and antagonizes the effects of acetylcholine, this allowing muscle contraction. Synaptic integration is a phenomenon in which multiple synaptic potentials congregate within a postsynaptic neuron. The neuron integrates these signals together to give action potential output. A method of measuring synaptic integration is through EPSP summation. EPSP summation has mainly two types of summation: spatial and temporal. Spatial summation is the adding of EPSPs that are generated concurrently at different synapses on a dendrite. Temporal summation is the adding of EPSPs that are generated upon the same synapse if they occur in rapid succession with little to no delay between them within a span of 1 to 15 milliseconds from each other. The figure here shows the various types of EPSP. In A, it is a simple scenario where an action potential from the presynaptic axon induces a small singular EPSP at the postsynaptic neuron. B depicts the spatial summation of EPSPs in which there are multiple presynaptic inputs on various synapses on the postsynaptic neuron. The adding of these EPSPs gives rise to a larger EPSP output on the postsynaptic neuron. In C, temporal summation is shown where the presynaptic axon fires several rapid action potentials quickly, resulting in EPSPs that add up together as they are being triggered. Despite having several EPSPs on the dendrite, the depolarization still might not be sufficient enough to generate an action potential. The ionic current that enters at the site of the synapse still has to spread across the entire dendrite through the soma and arrive at the spike initiation zone to depolarize it beyond threshold levels to initiate an action potential. This can be seen from the graph shown in the diagram. When the current is first injected in the dendritic cable, the amount of depolarization is at its highest. However, as the current travels along the dendrite, it slowly dissipates across the membrane. This dissipation of the current is highly dependent on dendritic cable properties. An index for determining this is the length constant, which is represented by the symbol lambda. This is defined by how far the depolarization can spread down across a dendrite or an axon. A larger lambda will mean the depolarization can spread further across the dendrite, and a smaller lambda will mean a shorter distance in which depolarization can spread. Lambda itself is dependent on two factors: the internal and membrane resistance. The membrane resistance depends on the amount of open ion channels. A higher number of open ion channels results in a lesser membrane resistance. If there is a greater number of open ion channels, the synaptic current, which prefers to take a route with lesser resistance, might leak out of the membrane, resulting in a shorter distance in which the depolarization can spread. As such, lambda increases as membrane resistance increases with lesser open ion channels. For internal resistance, it is dependent on the diameter of the dendrite and the electrical properties of the cytoplasm. As the diameter increases, the internal resistance decreases as the current would tend to flow inside the membrane more. Therefore, lambda will increase as internal resistance decreases. Ultimately, for a large lambda, one would prefer a high membrane resistance and a low internal resistance. In contrast to EPSPs contributing to the action potential output of a neuron, inhibition also occurs to inhibit the effects of the EPSPs through the effects of IPSPs. As mentioned earlier on in the video, inhibitory synapse channels are permeable to chloride ions and they bind to GABA and glycine neurotransmitters. The flow of the chloride ions act to bring the membrane potential closer to -65 mV, which is the equilibrium potential of chlorine. If the membrane potential was less negative than -65 mV, the hyperpolarization would be triggered. In the following diagram, the effect of IPSP in reducing the size of EPSP is seen. Initially, the neuron receives the excitatory synapse, depolarization occurs and spreads across the dendrite towards the soma. In A, the membrane potential of the soma was still higher than the initial membrane potential of the dendrite before the EPSP was induced. However, upon receiving the inhibitory synapse in B, the inward movement of the chloride ions, which is equivalent to the outward flow of depolarizing current, and the membrane potential is brought back to its initial levels. This inhibition is known as shunting inhibition, and taking the analogy of the garden hose in the video of action potentials, it is equivalent to cutting a large hole on the hose to allow water to flow outwards. Shunting inhibition greatly reduces membrane resistance and consequently leads to a smaller lambda, reducing the likelihood of action potentials firing.